Today what I'm going to talk about is a research paper, a review article, talking about the origin of the new coronavirus that's wreaking havoc everywhere, and essentially asking the question, where did it come from? And as previously mentioned, this is a review article. So this is summarizing around 30 different research articles from different fields and different types of research. And I'm going to talk about many of those. But first, I just wanted to give you an introduction to me and kind of my scientific past. So I'm originally from Virginia, and where I grew up. But after college, I moved to New York and stayed with my grandmother here in this nice, cozy New York City apartment. And during that time, I also luckily found a career in scientific research. So over the past 10 years or so, I've been studying and working in a lab. And so this is, well, let me switch to the pointer. And so this is me in the lab, and I typically study things related to DNA. And right now I'm studying how DNA is organized in a cell. So as you all might know, DNA is really long. So if you took a single cell and stretched out all the DNA, it would actually be much taller than me, about the size of Michael Jordan it would be. And all that DNA has to fit in a very tiny cell, and it organizes itself in different ways. And that's what I'm currently studying at Rockefeller University. What I'm curious about also is how DNA and other genetic information give rise to evolution in different traits. And I'm going to be talking about that in light of the viral evolution of the coronavirus. Okay, so this is me currently with my partner in our cozy New York City apartment, trying to maintain our mental health by doing some yoga. I've also been doing some sewing projects as well. And I don't know if y'all can see this, but there's a cookie inside of this mug. So we've been doing a lot of baking, and a couple of cookies a day is also helping keep our spirits up during these trying times. Okay, so now to the research. So as Jeannie mentioned, the name of this research is called the Proximal Origin of SARS-CoV-2, or as I'll refer to it as the new coronavirus. And this question that they're asking is essentially, when did the new coronavirus, as pictured in a cartoon here, become deadly to humans? Again, this is a correspondence or a review article, but there are still many different authors from different parts of the world that have collaborated together to write and critically think about this topic. But you also notice that something quite outstanding here is that this article has been viewed 4.2, almost 4.3 million times by different people throughout the world in a very short time. And one of the reasons for this is because of the implications that this article talks about is whether or not this new coronavirus was a result of bioterrorism, which has been debunked, and their rationale I'll present to you today, but also I want to present to you the rationale of where they think it might have come from and became deadly to humans. I also want to kind of tell you the techniques that that allow us to track genetic changes. So that's also part of the goal of this talk. And I just want to reemphasize that even though the authors didn't do the research, they did do a lot of, there are many authors, and they're coming from different parts of the world, a couple within the United States, but there's also some in Australia and in Edinburgh University of UK. But a lot of the research has been done in China, but also different parts of the world. So this is really a collaborative event, and just highlighting that in science it is such a important thing to cooperate. So before we get into talking about the actual research and the discussion underlying it, I want to go over a few key terms. So I know the central dogma was reviewed in this series last week, but I also want to review it just briefly. And then also I'm going to be talking about important region of the coronavirus protein called the receptor-binding domain. I'll also be talking about this technique called sequence alignment and how that is used to watch the evolution, how we track the evolution of the virus through that, and using this idea of a molecular clock. Okay, so so we have our virus here, and inside the virus there is this gene material or genome, as I might refer to it sometimes. And this in the virus is an RNA molecule. In humans, this is a DNA molecule, but the virus has RNA. And this material is the founding material that gets started and tells us the information how to build the cell. This material then gets transcribed, essentially copied, like you would just transcribe a note, into what we call messenger RNA, which is just an intermediate stage that allows the cell to take this information and then convert it into protein, which is a really important part. And this is called translation. And the reason this is called translation is because it's, in some sense, a different language, even though it's directly based off of this RNA language that it's coming from. And this language has what we refer to as different kind of letters, or we refer to as amino acids. So these are just abbreviations for those amino acids. So you can see the YHTVS, they're just shorthand, and they're organized in this linear string. And these encode for the very functional parts of the cell, the proteins that execute the tasks. So for example, y'all might have heard of a spike domain or spike protein. And so this is important for the infectiousness of the virus. And I'm going to talk a little bit more about this protein and its different domains. But before that, I just also want to remind y'all that this genetic element here, the RNA of the virus, it needs to be able to replicate itself. And during this replication, this can sometimes cause mutations. And these mutations generally happen randomly, but essentially like making it in an error. And these errors can be propagated and continued on into the next virus, passed along. Okay, so we have our protein words made out of our amino acid so-called letters. But these can be expanded on, just like we can create words into sentences and give it more information. And so for proteins, how this works is that they, this linear organization of the amino acids then can fold into different structures and shapes that allow it to give it different tasks within the cell. And this can be even more complicated and give more context to how this domain works with different parts or different domains of a protein, or with different, even more proteins. And so what I'm showing here, this green one, this is actually the receptor binding domain that's found in the coronavirus, the new coronavirus. And this domain is really important for its ability to interact with cells that express this ACE2 receptor. So these cells include the human lungs and other different tissues, but the lungs are one of the most prevalent one that express this receptor. And this receptor binding domain, as the name suggests, helps it bind to the cell and then allows it eventually to interact or to enter the cell and become infectious. So the way we think about these different shapes are kind of like this very classic toy in which you have shapes that correspond to different different receptors. So, for example, this one here would fit nicely in this small hole, whereas it might fit in these other ones, but it might not be as snug or as compatible. So it might fall out more easily. So that's kind of how we think about proteins and their shapes and how that informs different interactions in the cell. Okay, and so now the technique that is most used and talked about in this research review is this technique called sequence alignment. And this can be done with the RNA sequence, the genes, but what we're going to talk about is just the protein sequence. And so the goal of this is just this, it's similar to a search function or a find. If you want to find all the things that are similar, they don't necessarily have to be the exact sequence, but you can see that you want to line the sequences up so that they are most similar, even though there are some differences here. Yeah, so essentially just finding things that are similar or related to each other. And one of the things that this does is allows us to identify those amino acids, those that stay the same. And those might give us some insight into the structure and what kind of structures are important for the protein to create different shapes, but also what things that the virus might need to always maintain. Some other ones that are more mutated and changed are often those that can evolve and take different shapes, and those also might be important for understanding the evolution of the protein in the virus in general. And so what we can do is use this alignment technique to essentially map the what we call as a lineage, but it is essentially trying to understand who came first and then who gave rise to the next one and the next one, just like we think about our great-grandparents or grandparents and then all the way down to a child. So the way this works is essentially we take those sequences that are most similar and those that we think are the most distant ancestors or relatives. And then we look at the difference. The next one we'll map is the one that has only a slight difference. So in this case, this S changed to a T, but otherwise it's the same sequence. And so this is likely to be the next step of a mutation that occurred, but this T is maintained throughout the rest of these, is passed on. And then other mutations can build off of that and create more changes. And so this is the way we track time, essentially, in a genetic or in a genetic lineage, because proteins are ultimately coming from the genes. Okay, so now we reviewed these key terms. We have the central dogma, which is essentially going from genes to proteins, and these proteins are functioning in the cell and have like tasks. And the one protein domain that we talked about of the coronavirus is this receptor-binding domain, which is really important for its ability to interact with some of the cells in humans in the ACE2 receptor. And then we talked about the sequence alignment, which is essentially just identifying who's related, who's in the family, who's come into the cookout. The next is a molecular clock, using this sequence alignment to essentially trace lineage and see who came first, who's the intermediate, and who's the most recent one. Okay, and because this is a review article that's encompassing many different types of research, I'm just going to go over the different styles of scientific reasoning, because there are many types ways to create scientific knowledge. So one of the first ways is this categorization and classification. And in this paper, essentially what that means is collecting the genes and the genomes of different viruses. Okay, so sequencing the genes and compiling them. And then there's the historical based evolutionary reasoning. So these are different ways of thinking that have been established in the evolutionary field that give us ways to interpret the data that I'll talk about. And then lastly, there's this hypothetical modeling, which is essentially taking computer math equations that build off each other and use different scenarios to calculate different possible things. So you've probably heard this in the context of modeling for the amount of cases of coronavirus that are going to happen in different populations. And those can be changing depending on the human behavior. What I want to talk about is hypothetical modeling of proteins and the structure of proteins. But first, I just want to review that the overall goal of this study was to determine how the new coronavirus emerged and when it became deadly in humans. Okay, so the authors discussed three possibilities. So one is that the coronavirus originated in animals and that the current deadly form to humans originated in animals and jumped to humans and then began to spread from human to human, and it was already quite deadly. The alternative hypothesis is that that it began in animals in a kind of intermediate phase where it wasn't as deadly, and it jumped to humans, and then it passed from human to human, and that allowed it to adopt its more deadly form. And lastly, what people have also talked about is that it's possible being made in a lab. And this has largely been debunked as a form of bioterrorism. And I'm going to talk about the reasons in the paper that they give for this rationale. Oh, so, but first I want to talk about it's starting in animals. And so a lot of people have heard of the coronavirus in relationship to the bat, because the bats do have a lot of coronaviruses and different types. But there's also this other animal that I didn't know about until I started researching for this talk. And I just wanted to, step back and take a poll and see if anybody else recognizes this animal and knows what kind of animal it is, or this is really, everybody's really good. And so the possibilities are that it's an anteater, it's an armadillo, it's a pangolin, or it's super cute. And a lot of people are nailing it right. C is the most accurate. It's a pangolin, which is a small mammal. It is also a, okay, I have to, okay, share results, sorry. I can't get the poll out of here.
All right, I will take it off.
Okay, so, but they're also super cute too. So that was also a valid answer. So I spent a whole afternoon. Now I can't move to the next slide. So I spent a whole afternoon looking at pictures of baby pangolins, which are essentially a real life baby Yoda. And so they're super adorable. But they also unfortunately harbor the new relatives of the new coronavirus. And so how the data that the research is talking about was collected was that the cells from different animals, including from bats. So actually, from bats, it was taken from bat droppings, and the sequences that most people know from bats actually come from about 7 years ago, whereas the sequence that come from pangolins actually come from very recent. So within the past few months, there was a pangolin that died from coronavirus-like symptoms, and that was in a wildlife refugee. And they took the cells and sequenced their material, their genetic material. And this has an asterisk here because sequencing the genetic material from viruses from animals is very difficult. When this genetic material is sequenced, a lot of it is actually coming from either the bat or the pangolin itself. So very little of it is actually coming from the viruses. And thus, there is a really incomplete catalog of all the viruses that have been sequenced. But the ones that have been sequenced, what we've done with that, or what they've done with it, is they've used that to make sequence alignments and determine how related different viruses are to each other. And so the general overview is that there's a highly related bat coronavirus, so 96% to the new coronavirus. So overall, it's very, very similar. But there's also a pangolin coronavirus that is 91% similar. So it's also very highly similar, and it has a really interesting, unique signature that I'm going to talk about in a second. And then the SARS coronavirus, so this is the 2003, generally referred to just as SARS. This is less related, about 80% related, okay? And then also this receptor binding domain has the most changes. Okay, and so the data that they present in the paper is essentially what this is. This pink bar is just a cartoon of a protein with all of its amino acids. So this is what they're showing is the spike protein. And these numbers refer to the amino acids that happen in just right next to each other, just in this kind of line. And what you can see are these blue lines here, and those are indicating important changes in this receptor binding domain. And so if we zoom in a little bit closer, what you can see is what they have is the human SARS-CoV-2, the new coronavirus. They have the bat coronavirus that's 96% similar, and then the pangolin coronavirus, and the original SARS coronavirus, and two more distantly related ones. And so what you can see is that the bat is very closely related to the new coronavirus, but there are some key amino acid changes that exist between the bat and the new coronavirus. But so this F and this L, this Q and this Y, this S and R, and these other ones here, but these also are found in the pangolin. So they're actually very, very similar to the ones that are found, and they're known to be very important for the binding of the coronavirus to the ACE2 receptor that allows it to infect the lungs and other cells. So even though the bat coronavirus is the most related to this one, the pangolin one has this really unique signature that's really important for the infectiousness. So this indicates that the new coronavirus likely comes from a natural source because of its high identity to the bat coronavirus and the signature found in pangolins that is already existing in nature. So this is one strong argument for it not being created in a lab. So I'm going to talk about just briefly this other site that you can see that there's some other important changes. And this is referred to as the cleavage site. And the cleavage site is just generally important for not the binding of the receptor of the virus protein to the ACE2 receptor, but instead the entry into the cell. But what you can see here is that this new coronavirus has this amino acid signature that none of these, not the pangolin nor the bat coronaviruses have. So this is likely something that more recently changed. And this has also been confirmed in experiments that it is important for the infectiousness of the coronavirus in humans. Okay, so now it's time to talk about the interpretation of this data. So those are the major findings. And the question is whether this virus originated, the deadliness of this virus originated in animals or in humans. And we already talked about that it's very unlikely that it didn't originate in a lab, but we're going to do another poll and see what everyone thinks. Okay, so this one wasn't as easy as the pangolin question. And the reason why is, I guess, because of that is because it's not clear. So that's one of the reasons why. Okay, sorry, I can't get out of this one as well.
Can you just X out?
Uh,
Oh, stop sharing. Here we go.
Yeah, okay. And so that's one of the reasons why people often write review articles, is because there are some unanswered questions in the field. So the authors suggest that this receptor binding domain, because it's so similar to the one that's seen in the pangolins, this part was already mutated in some animal. It could have been bats, it could have been pangolins, and then jumped to humans. And it's possible that this cleavage domain was already existing then too, but it's also possible that this cleavage domain made those important changes while transferring to human to human. And the authors say that more scientific data could swing the balance of this in evidence to favor one hypothesis of the other. So that means that there's still an open window on this question, and some of the things that could be used to answer this is if they find viruses from animals and that that do match it more similar, that would indicate this kind of hypothesis. They could also use sequences of viruses that come from humans before the outbreak started, and that might favor, that might shift it to this hypothesis. Okay, and so as we mentioned, this is unlikely, but we're going to also talk about some of the other reasons the author mentioned that this is not only unlikely, but it's very unlikely. Okay, so the one thing that we mentioned previously is that there's a natural signature. So from the sequence alignment, just the uncanny similarity between those found in nature really suggests that it is a product of evolution in humans or in animals, or starting at least in animals. But there's also this really important reason, is that because viruses, it's not very easy to isolate a virus, because there are also many different viruses within one cell or one sample. And so when we isolate viruses in the lab, we often add these DNA signatures to them. And these DNAs, or in this case, it would be RNA, sorry, but we add these genes to them that allow us to track them and manipulate them and make them very easy. And otherwise, it's really very difficult to work with those in edit them or manipulate them in ways that would be necessary for a bioterrorism agent. So all the sequences that have been found thus far lack any kind of signature that is found in the lab, both in humans or in animals. So none of those exist. Another reason is using modeling data for the protein structure and the interaction between the receptor binding domain and the ACE2 receptor. So what researchers found out is that the structure and the shapes of the receptor binding domain that are found in the new coronavirus, even though they're very infectious and make everyone sick, they're more like taking one of these little circles and putting it in this big hole where it doesn't really fit. It can fit there, but it's not really snug. And so they suggest if that somebody were to create a bioterrorism agent, they would create something that is more snugly fit, more efficient at doing its job and in becoming more infectious. Okay, and so the general conclusions are that the new coronavirus has a signature that's occurring in nature. It has the highest sequence similarity to a virus found in bats. It has a receptor binding domain signature of a virus that's found in pangolins. And this is quite recent, this virus infecting in the pangolin. It has a cleavage site that's also important for its function, but this is more recent, and it's not clear in what animal or in humans it originated. Okay, and then also the receptor binding domain is not optimized to bind for the human ACE2 receptor, suggesting that it's not engineered, and it also lacks any signature of lab engineering. Okay, and just to review, this paper used a couple of different methods. It used the categorization and classification. So that's essentially taking and sampling as many virus genes or genomes that we could find in nature. And one thing to note about this is that we don't have a complete collection of all the genes and genomes of viruses found in different animals. So there might be some that come to light that change our interpretations of this. Okay, so there's also historical-based evolutionary reasoning that we used. And one thing to note is determining the original ancestor. So this is often difficult, mostly because of this incompleteness of the genomes. And so this is something that researchers are still trying to do, is determine the original, most recent ancestor of this virus. And then these hypothetical models that I talked about, the protein structure not being optimal, these are being confirmed in living cells and using these models to predict those different, more efficient structures. And a lot of these, a lot of what is known from the predictions are turning out to be accurate. Okay, and so why is this kind of question important, other than knowing that it wasn't an act of bioterrorism? This research can help in many different ways. So we can know, for example, which animals can be hosts to the virus. So this could prevent future transfers of the virus from animals to humans, but it also could help us stop spreading the virus in animals. So that might be an important way to stop spreading the virus. But we can also track, using the same techniques, track the ongoing evolution of the virus. And we can essentially look how the virus is being transmitted from one population to another using the same kind of techniques of this sequence alignment. And then lastly, these protein models can also help give us information about what areas of the protein to target when we create drugs or vaccines. And so that's also very important to think about.